Okay, so today we are going to talk about antigenic variation in Borrelia, but also, I mean, this happens in many different pathogens, including like malaria, Plasmodium vivax, and syphilis, which is like T. palladium. So this happens a lot in many different organisms that infect creatures. And before we discuss antigenic variation, I have to do a brief review of, this will be a review for most of you, hopefully, a review on concepts of homology, because this is going to become important for this discussion on antigenic variation. So what is the definition of homology in biology? Nobody?
Shared something between different organisms, something either being gene, some sort of.
So shared's a good keyword. Lauren, you took my class. What did I say about homology? Oh, okay. Well, Lewis is essentially right. It's shared, but I guess a better way to say it would maybe be like similarity. So similar structures or similar, if we're talking about like molecular genetics, similar sequences, which are then going to lead to similar structures. And the reason the shared is a good is because most of the time we're talking about shared ancestry. So if you have two different things, X1 and X2, when we're talking about homology, these things are homologous, they're similar, and they're probably derived from a common ancestor, X0. So this is kind of like homology. So the classical examples are like the bones in your hand. If you look at like the bones in your hand and then you look at like a whale fin, right? Like you can see like the same patterns of like the digits in like the whale fins. So that would be like a homologous structure. Those bone shapes would be homologous. But homology doesn't have to just be about structures. It can be molecular sequences. That's the gist. So homology is like this broader concept, like things that are similar. Under homology, and especially with respect to like molecular genetics, there are some key terms which we talk about a lot: orthologs and paralogs. Okay, so these are, if you ever hear like homologs is kind of like a catch-all term that could refer to either of these. Homologs are things that are similar, but orthologs and paralogs mean something very specific in molecular genetics. So paralogs, does anybody know what that is? What does like parallel make you think of?
Something that shares like a similar function, but came from different places.
No, that's not quite correct. Paralogs are usually derived from genetic duplications. So the classical example that I always use this example is a human gene, or this is a gene of vertebrates. Is hemoglobin. What does hemoglobin do? Hemoglobin is a protein that binds iron, and the iron carries oxygen, and it's what is actually the protein that carries the oxygen in your blood. There's different types of hemoglobin. There's like alpha hemoglobin, beta hemoglobin, and there's some other versions. So a classical example of a paralog would be that in one organism, say a human, a key thing here in paralogs is in one species, so we're talking about Homo sapiens here, we have, I think it's like more than 4 copies of hemoglobins. Now, these are all homologs of each other. So the alpha hemoglobin is a homolog of the beta hemoglobin, but it's more accurate to say that they're paralogs, because the paralog means that they're really derived from a genetic duplication event that happened, and there are multiple copies in one species. That's what paralog means. So if you have paralogous copies, that means you have more than one copy of one particular gene in a genome. Why would you do this? Why? So if we're talking about paralogs, and I just said that paralogs are kind of like genetic duplications of genes. Why would you do that? Why would an organism in any circumstance want to duplicate a gene? Okay, elaborate on that. So Lauren is saying if it's an important gene, you want more than one copy. Why? Yes, so that's right. Let me try to sharpen the language a little bit. It builds redundancy. Redundancy is useful in biological organisms for a couple reasons. One is it permits evolution and specialization, because what happens when things evolve is they accumulate mutations. Most of those mutations are damaging. Most of those mutations are deleterious. So if you only have one copy of a key protein that's essential, it cannot evolve, because if you mutate it in a bad way, you will die. Does that make sense? But if you have two copies of an essential gene, A and B, you can mutate B all you want, and A is still going to be functional. So you're not going to die. So essentially, redundancy permits the evolution and specialization. So if B is mutating and evolving, that can cause scenarios where all of a sudden B might essentially become useful in a particular context. And that would be then specialization. So redundancy, paralogs sort of permit evolution in a sense that they build in redundancy into systems, and they permit evolution. Okay, the second term, what I'm going to talk about now more, is orthologs. Orthologs are different from paralogs in that paralogs were duplicate copies in one species. Orthologs are the same gene in different species. Okay, so if I'm talking about hemoglobin alpha from Homo sapiens, and I try to make a comparison to hemoglobin alpha from chimps, the sequences are not going to be exactly the same. Well, I mean, maybe they are, but if I was talking about hemoglobin from like a mouse, there are for sure going to be some differences. And when we talk about these genes in comparison to each other, these are a pair of orthologs. They're the same gene, it's doing the same function, but it's from a different species. So if we say the ortholog of hemoglobin alpha from Homo sapiens might be a little bit different from the ortholog of hemoglobin alpha from chimps. So paralog is duplications within one species. Ortholog is the same gene amongst different species. Okay. Another term while we're on this topic that's worth discussing is analog. Analog. What would be an analog? How is that different?
I think their function is similar, but they're
Yes, that's exactly right. Function is similar, but they're totally different things. Different things. And what I mean by that is they don't share a common ancestry. That's the key point, is there's no shared ancestry. So a good example of this would be like insect wings on a dragonfly compared to wings on a bat. Like, they're not even close to the same thing, but they're functionally, like, the purpose is the same. They're for flying. Okay. But they're not, there's no shared common ancestry. And analogs are evidence of a special type of evolution. There's a term for this type of evolution where you see the same structure appear independently in different settings. What is that called? Yeah, you're close. Convergent evolution. This is convergent evolution. This is a key term, an important concept, is that things that are useful evolve like multiple times. Not everything just evolves once. Like, things that are useful, you see those things reappear in different lineages. So wings, you see them appear multiple times. I'm trying to think of if eyeballs. There's probably convergent examples of convergent eyeball evolution. What else would be a good example of this? Maybe wings is the best example I can think of. This happens in molecular genetics all the time, though. Like, there's a protein that does X in bacteria. You can find usually a eukaryotic protein that also does X, but they might be derived from completely different sources. So this is also a common theme in molecular genetics. Okay. Now, again, just a little bit more context before we go into the discussion of antigenic variation. So this phrase, I have this in the notes, selection will favor pathogens that persist in either hosts or reservoirs. Why is that? Like, in this context of this class, we're always thinking about there's an organism that's infected, there's a vector that picks up that organism and transmits it to a new host or a new place. And what I'm saying is here is selection is going to favor pathogens that are able to hang around in these places. Why is that? And how does this relate to pathogenicity?
You can survive like yourself or even first, but you don't know.
Yeah, that's exactly right. The host is, again, I've said this before, the host is like the vehicle that the pathogen is driving around in. And the pathogen is going to be under great pressure to attenuate itself to not kill the host, because as soon as it kills the host, that's a dead end. That's a dead end for that organism. It's gone from the face of the earth, because if its host dies, it's done for. So there's going to be tremendous pressure for pathogens to persist in hosts and reservoirs in kind of like attenuated states where they're like not causing lots of problems, but they're sort of like not being cleared by the immune system. So antigenic variation, which we'll talk about today, is one of these key mechanisms that facilitates this behavior. It allows the pathogens to persist in hosts and reservoirs for long periods of time without being sort of detected and fully cleared by the immune system. Okay, so now let's start talking about antigenic variation. Let's just first dissect the term here. Antigenic. What would that be referring to?
The thing that gets recognized.
Which is called what? Yes, exactly. Very good. Antigenic is referring to literally to the antigens. So if there's a bacteria in a cell or in a host, we've discussed the immune system enough that we know that there's things on the surface of this bacteria. Oftentimes these are called OMPs. This is a keyword, outer membrane proteins. They sit in that outer membrane, and when this bacteria or pathogen or virus interacts with things, it's usually like a touching interaction. They're looking to touch, and the touch is conveyed by these outer membrane proteins, these proteins that are on the surface. And our immune systems build antibodies which recognize these proteins, which we call antigens. So we understand the first term, antigenic, referring to antigens, which would be the, for example, OMPs. What did that imply with variation? Ifan, what would variation mean? It's different. They're shuffling. They're shuffling, they're mixing, they're changing, they're varying these antigens. And then what's going to happen? So imagine this is a true scenario where you have an antigen in a pathogen. The immune system builds up a massive antibody response against those antigens. All of a sudden, you make some daughters, and in your daughters, you completely change the antigens. Are these antibodies going to now recognize the new antigens? No. So now you've effectively hid yourself from the immune response that took so long to build up to try to kill you off. So there's going to be tremendous selective pressure as these pathogens divide. Which one is going to have the advantage, right? Like if this pathogen has two daughters and one carries the old antigens and one carries the new antigens, which one is going to do better? The new one, because the antibodies won't recognize it. This one will get recognized. This one's going to die off. This one's going to survive. And there's going to be now cycles. There's going to be cycles where these ones die. Now a new antibody response is assembled to recognize the new antigen. And then what's the bacteria going to do as soon as there's a new antibody response recognizing this? It's going to change it again. It's just going to keep changing it. So you're going to get these cycles of, okay, build up antibody A, the pathogen changes its outer surface, that antibody is no longer useful, build up of antibody B, pathogen changes its surface, that antibody is no longer useful, and it's just going to carry on infinitely. You can just keep changing, they can just keep changing the surface. That's antigenic variation. So mechanistically, let's talk about how this happens. How would you build a system to do this genetically? What kind of a mechanism are you going to rely on? It's a good thought, mutation. It's a good thought, but yes, say it again. Recombination. So mutation's a good thought. It's on the right track, but let's talk about why is this not good enough, mutation? What's the big difference between like mutating and recombination? What's really the big difference between these two things? They're both mechanisms of genetic change. Yes, Recombination is literally like mixing chunks. Okay, so you're like, I'm going to take 50% of this protein and 50% of this protein. Now all of a sudden, new protein is 50% different. Mutation is, let's make a SNP, a single nucleotide polymorphism. Maybe that will change one amino acid. Is changing one amino acid going to be significant in terms of an antibody response? Maybe, but probably not as much as mixing gigantic chunks of proteins. Does that make sense? So antigenic variation is driven by recombination. Okay. So let's just to make sure everybody's on the same page. Let's discuss reviewing of homologous recombination. So what is homologous recombination? We now know what like homology is. So what's that part of it? Things that are similar or different? Similar. Okay, so things that are similar in terms of sequence can recombine. What's the classical example of this? Like that everybody learns in biology 1. Yeah, what happens in meiosis? What's the famous, there's a term for it, a famous term. What happens in meiosis? Crossover, right? So crossover happens in meiosis. And the way that it works is, let's say this is chromosome 1, and in creatures like us, we have how many copies of chromosomes? 2. So because we are diploid means we have 2 copies. Not all organisms are diploid, but we have 2. So we have like an X1, an X2, and these are the same. These are homologs, or maybe it'd be better to say paralogs. They're duplicates of the same thing in the same species. And because they're similar, we can get crossover events where what you can produce is, let's draw these with a different color, even though they're the same thing. After crossover, what happens is you can get what would be called chimeras, which might look something like, I mean, I'm perhaps bad drawing, but something like this, like where it's a mixture of both, right? And that's why this is literally actually the purpose of sex. Like, it's to induce recombination, because recombination is a massive force for innovation in the cell. It's how it generates new things, by mixing gigantic chunks of this with that, mix this with that in big chunks. So this is literally actually like the driving purpose of sex, is because there's a tremendous advantage to having systems that can recombine. Mechanistically, recombination happens at what we call holiday junctions. And I got Christmas colors here. Holiday junctions. Essentially what happens is, I mean, I'll do this quickly, like there's papers and papers and papers written on mechanisms of holiday junctions. But essentially what happens is you get what's called strand invasion. If you have 2 sequences of DNA and they're similar, this strand can come down and bind to this one, and this one can come up and bind to this one. And this is what's called a holiday junction. And this sort of intermingling of the DNA strands creates these scenarios where what results is recombination. So recombination happens mechanistically through these holiday junctions. The person who discovered it, the structure, I'm assuming their name was Holiday. That's my best guess. So with all this in mind, what's a requirement for recombination to happen? What are the requirements? I mean, look at what I've drawn out here. What's literally like a requirement for homologous recombination to happen? Homology would be one, so similarity, because that's what's going to induce the formation of the holiday junction in the nucleic acids. And this can be catalyzed by enzymes, but you need that homology. What else do you need? There's something more basic. Yes, very good. You need to have more than one copy. More than one copy. So you need redundancy. If you only have one copy, you don't get homologous recombination. So you need these two things. These two things are a requirement of homologous recombination. Okay. With that known, many bacteria, most bacteria, I would suggest, are haploid, at least in most of the phase of their life. Like maybe there's a situation where they become diploid or aneuploid in the situation where they're replicating their DNA, and for a brief time, their cell has perhaps two copies of particular genetic contents. But bacteria, in the large part, are thought of as haploids. How can we get recombination in pathogenic bacteria like Borrelia? Now, we just said the requirements of homologous recombination, you have to have more than one copy, or it's not going to happen. So how can haploid bacteria recombine? This is kind of a trick question. What? Combine with another bacteria. It's not a bad thought, but usually when bacterial sex happens, what are they passing? Plasmids. Plasmids is kind of like a key thing. So plasmids, if here's a plasmid, Lauren, what's the function of the origin? Yeah, it's where replication starts, because the origin site is a site that recruits the DNA polymerase, and the origin codes for copy number of plasmids. So a bacteria, if this is a bacteria, might have one chromosome, but it might have 60 or 600 copies of an important plasmid. Now, are you going to get a lot of homologous recombination if you got 600 copies of something? Yeah, you're going to have like a lot. So if I was to look at pathogens, look at their genomes, where would I expect important outer membrane proteins to be? Important antigens, where would they be? No, they'd be on the plasmids. They wouldn't. They probably would not be on these large chromosomes. They'd probably be in plasmids. Important things that you want to recombine, you want them on the plasmids. So many of these important genes we're going to find on plasmids. That's why I always say really interesting genes, you find them on plasmids. Okay. So let's talk about Borrelia now. Borrelia is the causative pathogen of Lyme's disease. It's really good at antigenic variation. It's gram-negative bacterium. It's got an inner and outer cell membrane, flagella, doesn't live outside the host. They've sequenced its genome, and there's a really interesting structure to the genome of Borrelia. Borrelia has a very strange genome. There's linear chromosomes, and there's essentially 21 extra chromosomal elements. So if you think about like E. coli looks like this. There's essentially like one chromosome and then maybe like some plasmids. Borrelia does not look like that. Borrelia is this very strange creature with, and you could maybe say this is a fragmented genome. It's got different sort of like chromosomes. It's got different sort of like circular plasmid things. And again, like, why might this be useful? It's probably because Borrelia wants a lot of recombination. Like there might actually be a utility to this fragmentation. Now, I'm sort of like, this is kind of like my hypothesis. I'm kind of like stretching beyond where the literature goes. Speaking off the cuff, I'm suggesting like this is probably useful for recombination. Now let's look at this. This is one of these, I don't know why it's doing this strange thing, but this is one of these chromosomal elements. Okay. So this is, it doesn't even matter what the gene is. There's this gene called VLSE. This is an important antigen. It's probably on the surface of Borrelia. And look what Borrelia has done. Borrelia has taken and made hundreds, I think this ... means like it goes on for a long time, hundreds of different copies of different sequence variations which match in homology to this part of this antigen. So it's just got long, what they're calling silent cassettes, which means these are cassettes that are not expressed. This is the one outer membrane protein that is expressed, because here's its promoter. And essentially what this does is, this is the thing that is expressed, but if there's any selective pressure, it's going to infinitely recombine in infinitely different ways with these hundreds and hundreds of copies of this little section that matches this region in the VLSE gene. And because these are silent, is a selective pressure in silent genes that are not expressed, that don't do anything, is selective pressure sort of high or low in those genes? So if you think about evolution, selective pressure means there's tremendous pressure to keep the sequence the same. When does that happen? When it's a really important gene, you keep it the same. Okay. These are genes that are never expressed. They're silent cassettes. So what's going to happen in the genetic regions? Really? Nothing? What about, so every time, okay, if you think the answer is nothing, let's go back to biology of polymerases. Every single polymerase, which replicates and writes DNA, has an error rate, okay? High fidelity polymerase is 1 in 10,000. A low fidelity polymerase is 1 in 1,000. I don't know what the polymerase for Borrelia is, but imagine the polymerase has to replicate this every single time it replicates a genome. What's going to happen to these sequences? Are they going to stay the same over infinite periods of time?
Well, no,
They'll change, they'll mutate. And the reason that conserved genes don't mutate is because if you get a mutation, you die. These genes, I'm telling you, these paralogous segments, they're not expressed. So there's no selective pressure to keep them the same. They're literally just mutating willy-nilly to whatever happens. And then when selective pressure tells the Borrelia, we need to change this outer membrane protein, a recombination might happen, and all of a sudden you've got some completely new sequence that's been totally mutated, inserted into this outer membrane protein. Now all of a sudden the outer membrane protein is completely different. Does that make sense? So in these silent cassettes, there's going to be no selective pressure. These things are going to be mutating. I mean, they're still going to maintain some kind of a homology, like some kind of a semblance, because the mutation rate's not so high, but they're going to be changing drastically. And then it's going to create a tremendous amount of variation that can be shuffled into this cassette. And this is the cassette, and it's just shuffling all the time. So it's always changing the surface to something else. Okay. And if you look at this figure, you find this same thing in, this is a chromosomal fragment of Borrelia. Here's a plasmid from T. pallidum, which is syphilis. Same kind of thing. There's an outer surface antigen. It's got these sites with a variable region that are duplicated, and it just shuffles through recombination. Same thing with anaplasma. Anaplasma we talked about in the tick lecture. It's a marginal, is I think a cow pathogen. The human anaplasma is Anaplasma phagocytophilum. I think marginal is the cow pathogen, but it's got these plasmids with MSP2. What do you think MSP2 stands for? Major surface protein, major surface protein. So just like OSP is outer surface protein, MSP2 is major surface protein 2. And here there's 4 paralogs of, it's showing you 4 paralogs of major surface protein 2, but only one of these is expressed. Only one of these is expressed at one time. Here's the one with the promoter. And there's constant shuffling amongst the different variable regions. Does that make sense? So this lecture is sort of mechanistically understanding genetically how antigenic variation happens. It happens through homologous recombination, and to have homologous recombination, you need many, many, many paralogous sort of sequences that have been duplicated in the genome. Okay, this happens in Plasmodium vivax. This happens in Anaplasma. That's kind of it. Do you guys have any questions about this? What's the question?
So you talk about the science cases and use that the important protein will recombinate with gene in the silence casing.
Yes.
Change the protein.
Yep.
So this is the homology recombination.
So what you saw was this thing in Borrelia that looked like this. And these sequences matched homologously to what we call a variable region. Perhaps this region encodes the part of the protein where, if this is the bacteria, it encodes the part of the protein right here that is actually like in contact with the antibody. Okay. And what I was saying is Borrelia has taken this part and duplicated it hundreds and hundreds and hundreds of times. And what did I tell you in the beginning of the lecture? That duplications permit what? They permit evolution to happen and change to happen at high velocity, because it doesn't matter if you change a sequence here. It doesn't matter if you change the sequence here. It's not really expressed. It's not going to hurt the bacteria at all. This is just DNA. This is almost just kind of like extra DNA. It doesn't matter if you mutate the extra DNA. So a bunch of mutations are constantly accumulating in these silent cassettes. And then when the bacteria knows it's now all of a sudden needs to change the site where that antibody's binding, or it's going to get killed off by the immune system, it picks one of these stochastically, randomly. It doesn't pick, it just sort of like randomly. Homologous recombination will happen with one of these. Now all of a sudden you have, let's call this 1, 2, 3, 4, 5, up to N. And let's say this is X. All of a sudden homologous recombination happens and X is switched out for any of these from 1 to N. Does that make sense? And they're all different because they're all acquiring constant mutations, and they're all shuffling around and swapping in and out. Does that make sense? So it's essentially like when you think of like natural selection and mutations, and kind of like Lauren was suggesting, that if a housekeeping gene, something that's crucial, like for instance, perhaps something that synthesizes peptidoglycan in the bacteria, if that thing mutates, the bacteria will die, cannot survive. But this is a special gene that's literally been programmed where maybe its function, perhaps, like imagine a scenario where this is the outer membranes, here's the cell wall, and let's say this is the major surface protein. And what I'm saying is this region perhaps encodes this region right here, which perhaps has no function, but it's sort of like the thing that the bacteria is offering up, like, look at this, look at this. And so there's no negative downside to changing it because there might not be actual function to it other than the fact that it's just sort of like a bait, a decoy. Like, look at this thing. The antibodies should recognize this thing. And then all of a sudden they recognize it and you just swap it out. And now all of a sudden the purpose has become not having any purpose, but perhaps the purpose is hiding from the immune system now by sort of presenting these constant decoys that it can swap in and out. Does that make sense? Other questions?